Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. David Cowan. Listeners, David is Professor Emeritus in the Department of Environmental, Analytical, and Forensic Science at King's College London, and he is joining us today to talk more about measuring peptides and proteins in anti-doping, including some updates on a PCC-funded micro-grant and some of the other excellent research projects going on at King's College London. And listeners, David was actually the guest for our very first episode of the Anti-Doping Podcast, and we are so excited to welcome you back to the show. David, how are you today?
Well, I'm fine, thank you, Mary, and thank you so much for inviting me back again.
Well, we are thrilled to have you here with us and excited to get some of these updates that are coming out of the research that you're working on. So I guess to start off with, we'll take a step back here for listeners who might have missed our previous conversation in episode 1. David, can you tell us a little bit more about yourself?
I have a background in pharmacy, and I guess that most of my career has been focused on the safe use of drugs. You know, we have wonderful drugs around, but often they're misused. And it's a very important area also to look at safe use. And that's led into sports drug testing. I became a specialist in using mass spectrometry for analyzing small amounts of substances, often in fairly complex samples like biological fluids. I then helped develop the Drug Control Center, where I became the co-founder with my boss, Professor Arnold Beckett. And that developed until we were awarded the 2012 London Olympic Games, and I directed that laboratory. My research interest has changed over the years from small molecules to these bigger molecules of peptides and proteins.
Fantastic. Well, we're looking forward to getting into some of these details. I know I mentioned in our introduction that we're really going to be focusing on measuring peptides, measuring proteins for anti-doping. So to set the stage for our listeners, can you talk a little bit about the context? Why is it so important to be able to measure proteins in anti-doping?
Well, I think the simple answer is they're prohibited by the World Anti-Doping Agency because many of them have pharmacological effects that would give an unfair advantage to an athlete who uses them.
And we mentioned that we're going to talk about measuring these proteins, but it's not always easy. And I'd love to get into some of the details here. So, David, can you break it down and share what are some of the challenges that may be unique to measuring proteins?
As you know, Mary, one of the interesting things in science is things become easy when you solve them. And dare I say, it's been the same with our work on peptides and proteins. But they are a challenge because it's a different class of substance from the small molecules that we've looked at over the years, drugs like amphetamine, that's less than 150 Daltons in mass, up to the somewhat bigger compounds now, like the growth hormone-releasing peptides, which are as big as 3,000 to 5,000 Daltons. The problem is looking at bigger molecules, and the mass spectrometry work has to be modified from the way we will look at small molecules. Being bigger, the instruments need to be somewhat more sophisticated. The way we handle the compounds is sometimes more challenging. Some of them are rather sticky and seem to disappear very quickly. And indeed, that's one of the problems we face when working with insulin.
Absolutely. And I think in the context of anti-doping, one of the other challenges is just this idea of whether something is exogenously administered and whether something maybe is an endogenous protein, how to detect and how to sort of differentiate between what might be considered doping. Is this true?
That is a huge challenge, but small molecules like testosterone might be endogenously produced. So it is with some of the protein hormones like human growth hormone, insulin. A number of them, the administered hormone is identical to that produced naturally in the body.
Certainly. And I know you and your team recently received a PCC micro-grant to actually conduct research related to the detection of insulin administration. So I'd love to talk more about this project. David, can you tell us a little bit of the background and why you wanted to do this particular project and what the aims are of this study?
If I may, I would like to acknowledge the support of the Partnership for Clean Competition over quite a number of years on protein hormones. They funded our work on IGF-1, on P3MP, and the most recent grant, as you say, is on insulin. We've looked at the insulin problem for quite a few years, and it really is a big one. Banting and Best discovered insulin way back in 1921, but we're still learning about this molecule. In sport, the World Anti-Doping Agency requires its accredited laboratories to detect very small amounts. And when I was lab director, I was often frustrated with the difficulty we were having to get down to the sort of concentrations that WADA required. And it then occurred to me that we need to investigate what's going on. Where is the insulin disappearing to after it's been taken out of the body? That was the reason why I thought it can't just disappear. It's going somewhere. So how might we find out just where it's going?
Certainly. And you mentioned very small amounts. To give our listeners some context, can you describe how tiny we're talking about here in terms of the concentrations of insulin?
Something of the order, it's sub-nanogram per mil and of the order of hundreds of picograms per mil.
Oh, wow.
Yes, I was talking to someone just the other day just about how sensitive our mass spectrometric methods are today. And we used to talk about a spoonful of sugar in a swimming pool, but now we talk about a spoonful of sugar in large rivers, like the River Thames in London.
I think that is a really good way to sort of help us envision the tiny concentrations that you're talking about here.
It is tiny, isn't it? Yes.
Absolutely. And I guess taking a step back for listeners who might not be as familiar with the chemistry and sort of the relevance of insulin for anti-doping, David, can you tell us a little bit more about why insulin is important to look at for anti-doping and maybe what any sort of performance enhancing effects would be of administering this in sort of a doping context?
Insulin is a very interesting compound, and I think most people will know it actually controls our blood sugar level. And it has some very interesting effects on changing the sugar in the body and making it available for the athlete who wants it to cheat.
So can you tell us what were the steps for this particular project in terms of what you've completed and where you're at right now?
We knew that when insulin leaves the body, it does degrade into smaller molecules. And some of them are well known. Insulin-degrading enzyme has been a well-characterized enzyme for many years. But we thought of some other molecules that could be degradants of insulin that hadn't been researched. And we did an in silico type of research where we predicted what the compounds may be. And then we did some preliminary in vitro work that is outside of the human body and actually were able to pick up, to identify some of these predicted compounds. But we wanted to go a bit further than that, and we put together about some 18 compounds. I should acknowledge at this stage that when I say we, it's my colleagues, Dr. Vincenzo Abate, Dr. Ivana Gavrilovic, and of course the person who does the work, the PhD student, Mrs. Siam Mendo, who incidentally won a PCC prize for her work on the gonadotrophin-releasing hormones back in 2021.
So it sounds like you've made some fantastic progress in terms of starting this project. What were then some of the subsequent steps or milestones?
You know, one of the things in science is one comes up with an idea, which is no more than speculation. And one's got to convert it into a hypothesis of what you're trying to do. And then being an academic, we have to get money to fund the work. And when you've got the money for the work, then of course you've got to do the work. So that's when the work really begins. And in the case of these degradants, having predicted what the compounds were, we had to synthesize them. Now, fortunately, our group does have some experience of peptide synthesis. And for some of the degradants, the work was relatively easy. Now, insulin consists of some 51 amino acids built up into two chains, an A and a B chain. And it turns out that the B chain is relatively easy to synthesize, quite simply because it's got just two cysteines. Now, cysteines are sulfur-containing amino acids, and they will tend to link up with each other. Very much like the amino acids in your hair that makes your hair curly or straight. And if you want to straighten your hair, then by simply putting a reducing agent onto the hair, that smelly stuff in the perm, that will cause the sulfurs to break apart and become free thiols. And you can then oxidize it afterwards, having straightened the hair, and it will stay straight.
Oh, interesting.
It's similar when we're dealing on the micro scale with synthesizing peptides. We've got to link up the sulfurs. Now, if there are only two, there's only one way they can link up. So that's easy. The problem is with the A chain, which has four cysteines. I'm sure the mathematicians in the audience will be saying, well, we've got a number of permutations that we can get from that, but they've got to be pairs. And if you consider the number of pairs you can have with four cysteines, it computes to 3. So we have 3 different isomers. So when we produce our molecule with sulfurs, that is not linked up to each other, they can join up in 3 different pairs. And we had to make all 3 of them. And that's the challenge.
That makes sense. And your group has actually successfully made the reference materials that you were referring to. So can you talk a little bit more about what that process was like? How long did it take? And was it relatively successful? You mentioned it's difficult, but what was the actual on-the-ground progress? Yeah,
like many areas of research, it goes in fast, slow, fast, slow. You think you've made it, and then you find the yield is tiny. You've ended up with 1%, which is not enough. And then you find it's rather impure, so you've got to make it purer. And certainly the work does go in different steps. The synthesizer is a semi-automated instrument where you tell it what amino acids you want to link up, you give it the raw amino acids, and it links them one at a time in a chain. Because they've got cysteines, you have to block the cysteines so they don't join up in the wrong way. And that was the major challenge, to get the right chemistries working. And Siam Memdo, the student, did some amazing work. She went to a conference to speak to a professor there who had done work on some of the blocking agents, who gave her the tip that what was in the literature wasn't quite right and she needed to modify the way she did it. And that was the breakthrough for the more complicated A-chain cysteines.
I think that really emphasizes the importance of having these conversations and talking to other experts in the field, because if you had to solve this problem without that extra tip, it might have taken even longer. You may never have gotten there.
It is so important, and why it's sometimes important to talk even when things haven't worked, as well as saying how good you were at making something work.
Certainly. Now, I think part of what makes this particular insulin project exciting is that, David, you don't necessarily know what your findings are going to be, but it just has the potential to have really important impacts in anti-doping. So can you talk a little bit more about this and what this discovery process has been like for you?
So we wanted to make these reference materials only partly for identification purposes, because with the powerful mass spectrometry techniques we have available to us, we can do a pretty good job at identifying semi-unknowns. But we wanted to get an idea of how much is present of the different peptides. And we've started doing work now on blood samples to start to see which of the degradants actually are present in real life. We've got one or two so far, hot off the press. This is where we are today. But we don't yet know whether that's going to be important to help us better detect insulins. And we need to get down to smaller and smaller concentrations, which again is why we needed the reference compounds, to be able to see what was stopping us looking at small amounts.
Definitely. And I think this move from just developing and working in the laboratory setting and reference materials all the way to working with actual, as you mentioned, real life blood samples is a big step. So what other considerations or challenges have you run into moving now to the blood samples?
Of course, when we look at pure reference materials, then you don't have all of the issue with the biological background to consider. Once we deal with, in this case, blood samples, then we need to consider, are we looking at whole blood samples, serum or plasma, very different materials in terms of the background we may see when one puts the sample through the mass spectrometer. An interesting thing with insulin is that we know that insulin-degrading enzyme is one of the ways that insulin is broken down. But it's also known that hemoglobin, the material in red cells, will break down insulin. And the reason for that is that hemoglobin, when outside of the body, becomes not a means of carrying oxygen, but actually becomes a reducing agent, which breaks down the sulfur groups. So these cysteines, these disulfide links that are joined up in one particular way, endogenous insulin, then becomes these artifacts or degradants where the A and B chain, which were linked by sulfurs, are split apart, and the sulfurs in the A chain join up in 3 different ways. So that's a big difference between working in the test tube with the pure material and working with blood samples.
So, David, what are the next steps then for this line of research in your group there?
Well, Siam Memdo, the student, is a bit of a perfectionist, so she wants to do a few more experiments, but at the same time, she's got to put a PhD together. She's a PhD candidate, well on the way to getting that finished. And then we certainly want to do some more work together. And the next steps will be to take these reference compounds, make sure our methods for analyzing them do go down low enough. We think they do. And then to use it on real-life samples, which we've started to do.
So, David, I know you don't just work on insulin there in the laboratory. Can you talk about some of these other proteins and peptides and how they all are linked together?
I guess the first of the peptide proteins that I worked on was IGF-1. And the reason for doing that was that it's one of the biomarkers, one of the 2 proteins that is used to be able to identify growth hormone administration, the administration of a hormone that's identical to our endogenous hormone. The IGF-1 molecule consists of 70 amino acids and is about 7.5 kilodaltons in size, so mid-range. And the concentrations that one finds in the body is something of the order of 100 nanograms per mil or more. Going up in size, we have P3MP, which is the largest of the hormones that I research on at the moment. And that consists of 130 amino acids, 130 times 3. It consists of 3 equal chains, which makes it 42 kilodaltons in size. And not only is it much bigger, but the concentrations are much smaller, typically of the order of 1 to 5 nanograms per milliliter.
Well, David, that sounds great. And I know we talked specifically about some of the projects you're working on. We went into the details of the insulin project, but I know substantial advances have actually been made, and you've made some important contributions here in the detection methods for IGF-1. So can you describe some of these key advances?
IGF-1 was a fascinating story because I had a PhD student working on that now many years ago. And at first he was very frustrated with me, having a chemistry background and not used to working at small concentrations. And he said, how can he work on this without pure reference material? And I simply said to him, well, did Banting and Bess have insulin before they discovered it? And it's part of the problems with research. Anyway, he made a big success. And then the Partnership for Clean Competition funded an excellent project where we were able to do an interlaboratory study to make sure that our quantitative measurements of IGF-1 were the same as those at University of Washington, in the WADA accredited lab in Germany, the one in Salt Lake City, and the one at UCLA. And once we had got similar results, virtually identical results, then we were very comfortable that we could reliably measure IGF-1 using liquid chromatography coupled mass spectrometry. And that now is virtually routine.
Certainly. And I think ensuring this consistency across labs is critically important. What was that process like? Was it difficult?
We still work with this great group of people, again, thanks to the support of the PCC. We have a big project working on the collagen bone markers, so not just P3MP, but some of the other markers. The great thing about the collaboration, we were actually working with a clinical lab under the leadership of Andy Hoofnagel at the University of Washington. And that's been so valuable to have the sports doping labs and the clinical labs working together. And that's made some fantastic publications.
I think those collaborations are critically important in anti-doping, like you said, to have those conversations, to bring in people with different expertise. And compared to the detection of IGF-1, these detection capabilities, as we mentioned, for P3MP maybe are not quite as far along. So can you tell us a little bit more about the current state of P3MP detection and where things are going from here?
An interesting challenge with IGF-1 was we start by splitting it up into smaller peptides. And then the second phase of the project, after we'd done our first interlaboratory study, was to go back and look at the intact molecule, which in theory should be easier, but it wasn't when we started the work. So as we became more experienced in handling these proteins, then we were able to do an interlaboratory study on intact IGF-1. It's not possible to look at intact P3MP. To date, it's simply too large for the current analytical techniques. So what we decided we had to do was to split it up. Now, Dr. Daniel Moncrief, who was the key researcher who worked on this project, and indeed an international prize winner supported by PCC, in her work, she first tried splitting the P3MP into its single chains. So from the 390 amino acids down to 130 amino acids, splitting the 42 kilodalton molecule down to 1/3. And the reason she needed to do that was 42 kilodaltons is remarkably similar to human serum albumin, which is present in vast quantities. And human serum albumin actually reduces the mass spectrometer's sensitivity. And she worked out we had to get rid of more than 99.5% of the albumin for the technique to work. Even when she split the P3MP down, not just to single chain, but to smaller fragments, and she was able to select which of the fragments would be the best candidates for the research work. The fragments have particular code names depending on where they are in the molecule, and it so happens that she looked at a larger one on one end of the molecule called T1 and one a bit further along called T5. T5 is the smaller one and has proved to be the most successful. Interestingly, T1 is unique to the human, whereas T5 is also present in the bovine and other species. And because it was actually available in cows as well, it was actually easier to get hold of reference material. It's been very difficult to get hold of authentic P3MP. It's just a few of the challenges.
Absolutely. And you mentioned that there is this technological barrier that you aren't able to look at the intact or kind of the whole molecule of P3MP. Do you think, David, that the methods will ever get to the point where we can actually detect that whole unsplit molecule?
Well, in science, we never say never, do we? We never say always. We're always somewhere in the middle. Mass spectrometry advances every decade to huge increases in sensitivity. I can't remember the exact number at the moment, but the theoretical limit of mass spectrometry is thought to be about 1,000 molecules. And we're way, way, way less than that at the moment. So we're going to get plenty of advances in sensitivity. Different approaches in mass spectrometry also allows us to look at larger and larger molecules. So I do believe it's simply a matter of time before we improve the sensitivity in that area. In terms of sample purification, one of the successes we had with P3MP was to use immunopurification. That is to get suitable antibodies to capture the P3MP, to be able to clean off the albumin, and then to extract the P3MP and work it up for the mass spectrometer. We have some other ideas about alternative methods of doing capturing without using antibodies. And the reason we want to move away from antibodies is they tend not to be that consistent over time, quite simply because this is a small area of work and manufacturers are the best people at making them rather than researchers, and the manufacturers find it's not in a big enough market.
That makes sense. And I think you've done a great job of highlighting some of the challenges with these projects. But I think it is so important to continue working on advancing this area of anti-doping and their sensitivity. So what impacts are you seeing, or do you see the potential to have in anti-doping for this kind of work?
I think as time moves on, there are going to be more and more peptides and proteins that are misused by athletes. As new protein-based drugs come onto the market, then that potential misuse will increase. And that is one of the reasons why we've just got to continue our research in improving our detection of these often very effective molecules. That is to say, they work in very small concentrations. So we need very sensitive methods to be able to prove administration. And as you pointed out, sometimes we've got to actually distinguish the naturally produced hormone from the administered one. And sometimes the differences are extremely subtle.
Absolutely. So what are some of these differences you can pick up on? You mentioned they're subtle, but is it the configuration of the molecule? What are you looking for to differentiate those?
With the insulin work, we worked as a model compound, a version of insulin called Lispro, where the lysine and proline are simply swapped around. Yet we can distinguish that from endogenous insulin. One of the other areas is whether they're D amino acids or the more natural L amino acids. That is, in effect, whether they're sort of left-handed molecules or right-handed molecules, D and L being simply mirror images of each other, and as such are extremely difficult to distinguish analytically. And a colleague at King's College London actually has some methodology for a different purpose that may well be applicable to anti-doping to distinguish D from L.
Well, David, I know you and your colleagues recently published a paper detailing a new statistical approach that actually can be used to detect growth hormone abuse. So can you talk a little bit more about how this approach works?
Well, I don't claim to be an expert statistician, although our professor of statistics, who led much of this work, thinks I know something about it. Mm-hmm. The statistical methods are quite advanced that are used. But using those methods, one can reduce things down into relatively simple ways, such as creating equations where one can come out with numerical values, where one can set limits to be able to decide those who are dopers and those who are clean athletes. The factors that one needs to consider are quite big when one's dealing with endogenous compounds, because we all differ from each other, and yet these figures are based on the population value rather than an individual value. So we've got to work out we call them scores, what are the scores that you get from the population who are not using growth hormone to distinguish from those who are using it. Interestingly, the World Anti-Doping Agency have now started the line of research to do longitudinal profiling to be able to look at individual athletes. And fortunately, our methodology using mass spectrometry for IGF-1 is going to be very helpful for that, which is also why there's a need to get a similar mass spectrometric method on P3MP.
Oh, very interesting. So in terms of these longitudinal analyses that are starting, do you anticipate that these molecules, the concentrations, or kind of what you're looking for are going to vary substantially within an individual over time?
Interesting question about the resting values in different athletes and sedentary individuals. We actually discovered on the UK growth hormone team, led by Peter Sonksen and then Richard Holt, that it did depend on demographics. It did depend on whether you were a resting individual or not. There was quite a significant difference in your IGF-1 values. What that means then is we need to know a little bit about the athlete. At the moment, the main thing we need to know is the age, because we know that the growth hormone score does vary with age, being higher in youngsters who are growing and reducing down to a sort of a plateau in older individuals. So one of the additional things we need to know is not only whether the athlete is a male or a female, but also their age to produce the appropriate scoring factor. Once we go on to an individual reference range, then we don't need to know that data because we're going to compare, and it's coded samples. We don't know who the individual is, just work with the sample code, and the World Anti-Doping Agency database links it to an individual athlete, unknown to the laboratory. What we'll then do is compare you with you rather than you with me. So you end up with a much smaller variation, and we can be able to detect much smaller differences to be able to distinguish you when you're not using growth hormone from you should you use growth hormone.
Absolutely. And David, do you have a sense of how reliable these approaches are?
When we first picked up growth hormone administration was actually at the Paralympic Games with 2 Russians. And it was slightly nerve-wracking at the time because the first time you put out the test, you believe that it's okay, you believe that it works, but is there something you hadn't considered? When the 2 athletes admitted misusing growth hormone, then we were home and dry. Then we were very happy. One of the things we did in the Olympics was to collect a second sample as soon as possible after the first. And we could actually see the drop in score from the first sample to the second sample, which also gave us more confidence.
Oh, that makes sense. So in the first samples, the scores were higher than because the athletes were using growth hormone at the time. And then when you took the second samples, you saw that drop in the score from the sample collected at a later time point. And that kind of indicated that they had stopped using growth hormone and were returning then to their normal levels.
Yes. With the longitudinal profiling, because we'll be able to see this pattern of multiple samples, it will give us that much more confidence that we're not falsely accusing an individual.
Absolutely. And I know science takes time, but things generally move pretty quickly in anti-doping. So what is the process, David, or maybe the timeline for taking a new approach that you develop, like this one, from a research article or a research study in an academic journal to being actually used regularly in anti-doping or starting to test it on real athletes in the field?
There is a certain, I'm going to call it hesitancy by WADA, wanting to make sure that any new test has been properly checked out and isn't just some researcher's one-off attempt at doing something. Because we recognize that, we also make sure we do things like interlaboratory studies to make sure that it's not just one laboratory that gets good results, but many laboratories can come out with exactly the same answer. And that's so important in anti-doping because it's truly international work. And if the person is picked up as a cheat in one lab, then it's quite important they get picked up as a cheat in another lab as well. Any new test has to be approved by the World Anti-Doping Agency, who will do quite a few checks to make sure the test is reliable.
Absolutely. And you mentioned that you were a bit nervous kind of putting this test out there for the first time, working with athletes. What was that experience like for you and your team when you actually got out there and you saw that those athletes admitted to doping?
It so happened that it was very important for the Paralympic Committee because they were being accused of not getting enough findings during the games. And because it was a world first, then they were very happy to be the people who were involved with it. Of course, as scientists, there's always an excitement when you get the world first on any substance. And indeed, I've had a few over my career that always makes one feel that the research was worthwhile. To prove that it works in real life is so important.
Absolutely. Well, David, you've shared a lot about your exceptional research with us today. Are there any points that you didn't feel like you got a chance to cover that you want to tell us about?
One of the great things about science is it helps to answer questions. So I feel I've been very fortunate to have a career that I have had to be able to contribute to the field. It's nice to have the idea. It's nice to do the work. Of course, it's even nicer to have the success. Sometimes it's a long journey. And the main thing is that you believe in yourself, that you believe you are doing the right thing, that what you're doing does help others.
Yeah, I think, David, science has such potential to have a huge impact to help people to really advance this world of anti-doping and ensure clean sport. And if our listeners are intrigued by some of the topics we covered today and they want to learn more, what is the best way for them to do so?
Well, I wouldn't want to hit them with the vast number of publications that are out there in the field. So I'm going to cheat a little bit and suggest they start by doing a Google search on me. And then from that, look at some publications of mine. And from that, find other publications and satisfy their interest and hope develop the interest further.
Well, listeners, definitely take a moment, get lost in the literature. There is so many exciting research studies out there by David and his wonderful colleagues in anti-doping. And David, it's been such a pleasure to have you here with us today. Thank you so much for your time and insights.
Thank you, Mary, for giving me an opportunity to share some of my interests with your listeners.
Oh, it's been a pleasure. And listeners, always wonderful to have you here with us as well. If you haven't already, it would be wonderful if you could take a moment to rate our podcast and leave a review on your favorite platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.